Hello. I want to thank the MRA so much. Thanks for the introduction and thanks for the opportunity to create this presentation because I was given a challenge by MRA to help explain some of the technologies that are being used in diagnosing melanoma. Of course, everyone who has melanoma wants to know how firm the diagnosis is. How was the diagnosis made? And to understand that, I think it takes a little bit more understanding of some basic things. And I hope that what I've done is to create a presentation that will set everybody up to be on the same page and understand what the strengths and the weaknesses and the limitations of our current state-of-the-art diagnostics are. So how is melanoma diagnosed? And I think it's really important. What we're going to talk about is a drill down into the ways that that melanoma is the ways that they are diagnosed. Let me. Okay, so what we have is what you can see with the naked eye. That's one way of diagnosing. But you can use a dermatoscope, so you can see more with a dermatoscope. You can see more with in vivo confocal microscopy, which I'll talk a little bit more about. Don't worry that you don't know what that is yet. You can see more with a traditional microscope looking at the biopsy. And you can tell more from molecular changes that have happened in the tumor. And so what we're going to do is drill down into all of those areas of information. So starting at the visible body level, melanomas occur on the skin, but they also occur in the eye, and they can occur in the mucosa, including the nose, the mouth, and the genital area. So you can have melanoma in a variety of places. If you start to look a little bit deeper down into the skin, you've got two panels here. One is the skin and mucosa, and the other is the eye. And each of those are independent organs. And so if you look carefully, I want everybody to understand the structure because we're going to start looking at real slides here in just a minute so you can see everything. And you need to know what you're looking at. So if you're looking at the top of the skin here, and here are the little hairs coming out, you've got the epidermis, which is this purple colored wavy sort of element of the skin, and that's comprised of a lot of skin cells. And then those are mainly keratinocytes, and the melanocytes live right at this junction. And underneath that is the dermis, and then underneath that is the fat, and you can see sweat glands, and you can see glands from the fat that release oil glands into the skin. And here, right here, is a melanoma. And it started as just a few melanocytes right here at the border between the epidermis and the dermis. And it started to grow, and it began to grow uncontrollably. And then it moved into the epidermis, and then it started to extend down and invade into the dermis. And that is the way that a melanoma progresses in the skin and the mucosa. In the eye, there's separate different pigment cells, and they're primarily in the iris, and so those can make a melanoma. That's the colored part of the eye. You can also have a melanoma in the ciliary body, and that's this melanoma there. And one of the more common places in the eye is the choroid. That's called also choroidal melanoma, and it's back here a little bit further in this part of the eye that's called the choroid. So now you've got a kind of an idea of where the melanoma can start. But what does it look like? So I'm going to start with a schematic, and we're going to start to think, and bear with me on this because we're going to take this slow and steady, but we're going to get you to the point where you can really interpret the pictures that I'm showing you of real life situations. So here's a schematic, and this shows one of those melanocytes. Now remember, this is the epidermis. This whole area right here are the epidermal cells. They're the keratinocytes. And the melanocytes live right here at the boundary between the basal layer of the epidermis, and down below here, it's not pictured, but that's where the dermis would have been. So this is that purple structure that you saw before. And what happens is the melanocyte reaches out, and it has these little pigment packets, these little brown pigment packets, and it distributes them to the keratinocytes, right? And here's what you see when you actually look under the microscope. Here's again a picture of the epidermis, and here's the dermis. And if you look right here, that's a little melanocyte that you can see right there. There's another one right there. So now we've shown what those things look like. How do you start to use that to diagnose a melanoma? And the way that we do that is we combine some molecular technology with cellular, these great histological slides. This is called histology. So how do we do that? Well, the first thing is that you start to look, this is called an H&E, hematoxylin and eosin stained skin. This is the stain that when you have a biopsy that is taken from your skin, it goes into formalin, it gets fixed, and it gets stained. The first stain they'll do is hematoxylin and eosin. And what you can see is you can see those little melanocytes right there and there and there, but they're pretty hard to see. Here's just a close-up to kind of give you an idea of what's happening with these stains. So I thought this was a beautiful picture. This is a picture where the nucleus is stained blue and the cytoplasm is stained green here. And these are the way that melanocytes look when they're in culture. And these are the little projections that they're attaching to the keratinocytes when they go. And what we have identified is that there's a marker that's called S100. It's a protein that's created on the surface of the melanocyte. And we've created antibodies that bind to that protein. And when you put an antibody stain on top of this histology slide, what happens is it stains them darker brown. It will allow you to see those melanocytes even better. But S100 isn't the only marker. Oops. There are other markers that are called MART1 is another one. And MART1 is stained by an antibody called melanA. And this is what you see. You can see that these are even easier to see. So one of the ways that you can help improve the diagnosis of melanoma is to use some special stains that will allow you to see what the melanocytes look like, the pattern, the amount of stain that they have on them, which stains are positive and which stains are negative. And that's going to be the first thing that people will use to determine if you have a melanoma, is those special stains. But nowadays, this is where I'm going to get into the meat, and hang in here with me. I'm going to try to take you through one step at a time of the molecular level information that you need to know to understand the latest diagnostics. So let's pretend this is a melanocyte. And in the melanocyte, there's a nucleus, and the rest of it we'll just call cytoplasm. Now, this is really complicated stuff, but we're going to keep it really simple, that you have a nucleus and a cytoplasm. And in the nucleus, that's where the DNA resides. That's where you have all of the genetic material that is made up of DNA. Now, DNA, it's kind of like the code book. So if you were to think about the original Bible that was created, people wanted to transcribe so that different people could get it, DNA is like the original book, and RNA is like the transcription of that book. It's the message of each protein transcribed into RNA. Now, if you want to translate the book, that's like taking the RNA into protein, right? And the proteins are where the action is, right? The proteins are what are responsible for helping the cell to really be alive. That's what allows the cell to grow, to divide, to repair itself, to metabolize things, to communicate with all of the other cells. And even, I mentioned this one, to die, because one of the important things that happens to normal cells is that if they start to accumulate mutations, they can do a thing called programmed cell death. It's like cell suicide, and they'll kill themselves so that they don't pass on that mutation on to the next generation and lead to a cancer. Okay, so what happens to create cancer? Well, in the case of melanoma, a lot of it is caused by sun and ultraviolet radiation. There's other things that can create mutations, but we know that sun is one of the primary drivers of mutations in the DNA. So what happens is that sunlight comes in and it goes through the cell into the nucleus and mutates the DNA. And when it does that, you can see it messes up the code. The DNA is now not exactly correct. And so when it isn't correct, the RNA isn't correct. And when the RNA isn't correct, the protein isn't correct. And when the protein isn't correct, you start to get mistakes. You start to get problems. The cell can't control its growth or its division. It can't control how it's metabolizing things, the oxygen levels that it needs, things like that. It can't repair itself as well anymore. It can't commit suicide if it needs to. And it doesn't communicate properly with the other cells. It starts to tell the cells to build more blood vessels to help with the defect in metabolism. It starts to tell them to grow out of control with it, for example. So the point is here that there are definite molecules that are really, really important in controlling cancer. And this is what has come to be known as the stepwise progression to malignancy. So it's a stepwise process. Any of these hits by themselves are not going to lead to cancer because the cell is very resilient and it can counteract it. It can have alternate ways of regulating things for a long time. But as you get more and more and more hits, you stepwise move towards a progression to malignancy that ultimately, when you've got enough of the right combination, you end up becoming a cancer. The cell ends up becoming a cancer. And this is what, this is basically a really fundamental, important thing to understand, is that every melanoma is a unique cancer, right? It's genetically unique. No two melanomas have exactly the same collection of mutations that have led it to become cancerous. Now, there are some mutations that are more common than others, but none of them are identical. So how do we know? So that's the background. All of that was background to get you to the point of how do we know something's a melanoma? And the real answer is you won't know until it spreads past the skin to other areas and leads to life-threatening complications. That's the true definition of knowing when something is melanoma. But as we all know, that's a little bit too late. Wouldn't it be nice to know a little bit earlier than that? And so that's where biopsies, special stains, and molecular tests come into play. They are all ways to better predict if a mole is actually a life-threatening melanoma. And none of these are perfect, as you'll see. So what is the general principle of diagnosis? I think before we start to talk about how you make a diagnosis, it's important to understand that melanoma exists on a spectrum. It's not like there is a melanoma, there's a benign lesion and a switch gets turned on and it becomes a melanoma. Because I just talked to you about it. It's a stepwise progression to malignancy. And so there are moles that are unquestionably benign, and there are melanomas that are unquestionably malignant. But in between those, there's some probably benign, there's some probably malignant, and then there's some that are really, really, really difficult to tell. So if it's on a spectrum, then how do you figure it out? Because the reality is, just because we can't tell doesn't mean there isn't a ground truth. And what we're trying to do with diagnostics is to get to the best answer we possibly can. Is it really benign or is it really malignant? And how do we determine that? And that's what a diagnostic is about. So when you see an atypical lesion like this, right? Now, this person makes a lot of funny nevi, right? So it's a really good question. We haven't seen their whole body. Is this really something that is pretty similar to a lot of other moles, or is this something that needs a biopsy? And so what happens? What happens if you elect to biopsy this versus elect not to biopsy it? Well, if you do, then it doesn't really change the point that either it's benign or it's malignant, right? You can elect to biopsy it and you can find out whether it's benign or malignant, or you can elect to not biopsy it and it's still either benign or malignant, whether you've biopsied it or not. So if you elect to biopsy it and it's benign, then what you've done is you've created an unnecessary scar, right? If you biopsy it, you elect to biopsy it, and it's malignant, then what you've done is you've detected the melanoma at the very earliest stage that you could have detected it. If you elect not to biopsy it and it's benign, great. No harm, no foul. It's no scar for the person, and it's a great thing. But if it's malignant and you elect not to biopsy it, then what you've done is you've delayed the diagnosis, because it's not going to go away. It's going to keep coming, and it's not going to just disappear on its own. You've just delayed the time at which you've diagnosed, and it's very probably going to give you a worse outcome than what you would have had if you had the earliest diagnosis possible. So nobody wants to do this. And in fact, even though you don't want to cause a scar, the consequences of having an unnecessary scar are much less than that of having a missed diagnosis. So what happens is that it favors the biopsy over the not biopsy. And that's where you're going to hear a lot of controversy about when do you actually do a biopsy? How many biopsies do you do? And how does that turn out? So what if you have a clinically atypical mole? This is one of those indeterminate lesions. You don't know, right? And let's say you biopsied it or you didn't biopsy it. If you didn't biopsy it, what are you going to do? You're going to monitor it, and you're going to watch it and make sure it doesn't change. And if it does, you're going to get in and you're going to have it biopsied at that time. If you biopsy it, then there's three possibilities. It can either be benign, or it can be malignant, or it can be indeterminate. If it's benign, then you're done, right? Nothing else really needs to be done. Your treatment is complete. If it's malignant, then you need to have maybe another, you'll definitely need to have a wider excision. You may need a lymph node biopsies. You may need to have some imaging done to see if it's spread to any other parts of your body. You may need some genetic testing of the tumor, and you may need some systemic, either immunotherapy or targeted therapy or something else. If it's indeterminate, then you're still in that place with what do we do next? And this is what I'm going to go into later. So if you do the special stains and it's really clear, that makes it really clear, and it's malignant, then you just go down that pathway. If it's really clear that it's benign, then you go down the other pathway. And that is where the special stains and FISH and CGH and the JEP test help you. Now, I just want to bring out there, and I'll talk a little bit more about this in just a second, but there's another technology that is out there now that I'm very excited about, and it's called in vivo confocal microscopy, or the other RCM stands for reflectance confocal microscopy. And this is a handheld microscope. It's basically, you put it on like a dermatoscope. I think a lot of people have seen dermatoscopes. You put it on, but it is able to send light down and receive the light back up as a microscope does. Just like when you see a microscope send light down and it comes back, it sends it down and it's able to see it at the cellular level. So what it is it's a non-invasive biopsy. So it's a way that you can biopsy the skin. You can look for whether or not the cells are benign or malignant or indeterminate without having to cut the skin. So I think it's a big progress. It's a major progress that's coming in diagnostics. So if confocal looks bad, then you're still going to have the biopsy to do all of these other things with. But if it looks really great, then you can monitor it. And that's the big advantage to confocal. So let's do a diagnosis. Let's go through it. Let's start with visual inspection. So here we go. Let's just take this mole. These are two moles that may come into a clinic, right? And they come in, and this one's a little darker. It looks a little bit more suspicious. This one doesn't look so suspicious. But believe it or not, both of these are very similar when you get to the cellular level. And I'll show you that. But let's say you come in with that and you say, hey, Mr. Dermatologist or Mr. Primary Care Provider, I'm worried about this mole. Can you take a look? And they look, and if they have a dermatoscope, that gives them a huge advantage, because I think that you can see here, this looks very smooth. This looks to the naked eye, this looks like it's probably pretty benign. It looks uniform in color. It's symmetric. The borders don't look that irregular. Everything looks great. But if you look at it with a dermatoscope, and a dermatoscope is just a magnifying glass that has, sometimes it has polarized light, and sometimes you use oil like you see here to allow the transmission of the light to be better. And together with that bright light and the magnification, you can start to see that the edges of these, this is not so uniform. And these are little, they're called pseudopods. And they're little bits of the melanocytes that are growing actively at the border and are expanding and are extending out. And it's a sign, it's a bad sign, it's a suspicious sign. It means that it's something that's happening in that mole. And so this then, if a doctor or provider ever saw something like this under their dermatoscope, they would definitely say, oh, this is clinically atypical. Now, if they have a reflectance confocal microscope in their repertoire, if they have that available to them, they can look at it. And I think you can see here, you saw those little melanocytes that I showed you. This is what they look like with in vivo confocal. And you can see these little yellow dots. These are the melanocytes with the nuclei, and then the bright color is where the pigment is reflecting back that light. And you can see that they're big and they're thick and they have a lot of branches, and this is abnormal. And so this shows those thickened atypical melanocytes, and this would be a case where the confocalist, that provider, that doctor would send it would go ahead and biopsy, and would do a biopsy like what you've seen before. Remember, this is the epidermis, this is the dermis, and what you start to see, these are those melanocytes, those clusters of melanocytes, but now they're not a nice layer of them or one little collection. They're a bunch of different sized collections, and the cells in there, you can't see them very well, but they don't look normal. And this would be called an atypical spitzoid cell, or maybe they would just say, this is a possible melanoma. So let's continue the diagnosis. Now you've got this pathology. What do you mean, possible melanoma? Hey, doc, I thought that if you did the biopsy that you'd be able to tell me, is it a melanoma or is it not a melanoma? What do you mean it's a possible melanoma, right? And I think that's where it's really important to understand that the microscopic eye, the eye of the pathologist looking through the microscope, is not that different than the naked eye looking at those two lesions that we saw by the naked eye. And so, for example, let's just compare some of those things. So the size and the shape of the mole is what triggers a provider to say, this is an atypical mole. It may need to be biopsied. At the microscopic level, the pathologist looking through the microscope says, the size and the shape of the cells that make up the mole are a little atypical. But just like there's variability in the size and the shape, and where is the cutoff of what's acceptable and not acceptable, the same is true at the microscopic level. The naked eye, the dermatologist might say, color and color differences of the mole make it suspicious. Well, a pathologist might say the amount of pigment and that the cells are producing is the problem. But again, that's variable, and it's not black and white, right? It's a spectrum. The dermatologist might say, ooh, this is changing or growing. This mole has changed since the last time I saw it. That's a suspicious sign. Well, at the microscopic level, you can tell how many cells are in the act of dividing. Those are called mitoses, and you can tell how many are doing it. And that's another measure that they use. But again, how many mitoses does it take before it's too many? And it's a sign that it's actually become malignant. There's some gray zone there. And maybe it looks red, or there looks like there's some color that's changed, that's been eliminated. That's a sign for a dermatologist that maybe your mole is going bad. Well, the cellular part that is the corollary of that at the cellular level is if you see inflammatory cells coming in, or you see fibrosis that's changed. And so even if this doesn't all make that much sense to you, the bottom line is that the magnified eye has more information, but it is actually just as subjective as the naked eye. Having a pathologist look at it, that's why there is so much discrepancy between one pathologist to another, just like there is from one provider to another about what is in that indeterminate area. So let's keep going. let's add some other special stains maybe that the pathologist might have in their portfolio that they could try. There's a lot of new stains, and the really cool thing about these stains is that they have been identified by looking at the cell and the proteins that are important for identifying when a cell has become malignant. So, for example, Ki67 is a stain that marks whether or not there's cellular division happening. So if you stain with Ki67, you're going to be able to see better how actively dividing is that cell, which is a sign of malignancy. Similarly, one of the most important senescence genes, a cell that keeps it from growing, is CDKN2A or P16. And that, if it's missing, that's one that can also be a clue for the pathologist that this is a malignancy. So again, I want to reiterate, every melanoma is genetically unique. They all have different problems. And so even though you've got a nice panel of stains, and these stains will allow you to mark abnormalities on the cells, not all melanomas have each one of these abnormalities, and you don't have stains for every possible abnormality. So what does that come out to mean? It means that even though it's a really good piece of information to have, it sometimes isn't able to tell you whether or not to prove that this is a melanoma. So if special stains aren't good enough, then what's the next step? And that's where we get into the first of the molecular tests that I'm going to talk about, which is called FISH for short. It stands for fluorescent in situ hybridization. Fluorescent means that the markers that we're going to look at fluoresce. So you look at them in a dark room under a microscope so you can see the light, and you look at it with a black light that stimulates them to fluoresce. And so that's kind of cool. I'll show you a picture of that in a minute. And in situ means that it's actually in the tissue itself, like what we've looked at, that histology, it's in the tissue. And hybridization just means that it's binding to the tissue. So again, let's go back. We have here's your melanocyte, and here's the nucleus. And in the nucleus, you have the DNA, and the DNA is packaged into the chromosomes, and the chromosomes are where the DNA actually is. In the case of melanoma, the two chromosomes that are most important for FISH are chromosome 6 and chromosome 11. And so with FISH, what you're determining, you have these little stains, these little probes that bind to the DNA. They match up with the DNA, but they bind to DNA that is important for controlling melanocyte growth. And so if they're out of whack, then if you have too many copies of them, or you have too few copies of them, then what that tells you is that that cell is not going to be functioning completely normally, because those proteins that are made from those pieces of DNA are important for regulating the growth of the cell. So in FISH, gene copy numbers are detected by the probes that are labeled with fluorescent markers, and we have one up here on that this is called the P or petite arm, and this is the Q arm. The RREB1 probe is important for that gene that we know helps control melanoma. We bind one here, CEP6, another one here, MIB4, and another one here, CCND or cyclin D1. And these 4 probes can be applied all at the same time. You break open the cell so that the probes can get in. They bind to the chromosomes, and you can start to see whether or not there's more than one copy or less than 2 copies. And I'll show you an example. If you really want to look, you can look at this Coolaboo. There's some places you can look at more detail if you want. So this is what they actually look like. This is what the pathologist who's making the diagnosis looks like. So to orient you, this is a piece of the tissue at high magnification, and all of these little blue circular things are the nuclei of the cells. They're stained with a dye called DAPI, and that makes them blue, and that tells you where the nuclei are. And then what you can see here is a normal cell. This is a normal nucleus, and as you would expect, every every cell nucleus is supposed to have 2 copies of every gene, one from your mom and one from your dad. And so these are the genes that I just told you about that are important for regulating melanoma. And in a normal cell, you have 2 yellows, you have 2 reds, you have 2 blues, and you have 2 greens, and it looks perfect and there's no problem. Now, if you have an abnormal cell, this is an abnormal, a nucleus from an abnormal cell, and you can see that there's only there's 4 blues, 1, 2, 3, and you can barely see that blue one right there. And that's another problem, right? Because there's some subjectivity here. You can't, it depends on how good the pathologist's eye is. But there's 4 copies of the blue, there's 1 copy of the yellow, 1 copy of the red, and 3 copies, 1, 2, 3 of the green, right? Now, if you look at a melanoma is even more dramatic, right? You've got multiple copies of multiple genes and maybe even a complete loss of other genes. And again, what I want to point out here is that every melanoma is genetically unique. So FISH identifies abnormal numbers of multiple genes, 4 different genes simultaneously. But again, it's subjective. It's the dermatopathologist or the pathologist, the molecular pathologist has to look at it and make a call. How many cells like this do you need for it to be called a melanoma? How many different colors in one nucleus do you need to call it a melanoma? It is incredibly informative when you see something like this and all of the other stuff is still indeterminate for you to say, this is probably a melanoma. But it doesn't detect the specific mutations, and it doesn't always prove it's a melanoma. So there is still an indeterminate category. So that takes us to the next kind of test. It's called CGH, Comparative Genomic Hybridization. And the A stands for Array Comparative Genomic Hybridization. And again, this looks like a complicated slide, but I'm gonna take you through it slowly. So if you want to watch a video, this is a really nice video, and there are other videos out there that can explain it in more detail if you're interested. But let's just start with that. We're trying to make the diagnosis. We've got an indeterminate mole. We don't know whether or not it's really a melanoma. It's a possible melanoma. So what you do is you take the tissue block, the biopsy that's been done, you take that tissue block and you take some cells out. And you take similarly some normal control cells that are just a standard normal DNA. And what you do is you break open the cells and you pull out, extract out, and purify the DNA out of those cells. So now you've got DNA from your questionable biopsy that we don't know what it is yet, and you've got good, normal, healthy DNA. Then what you do is you take the questionable DNA, the possible melanoma, and you label it with, you just put markers in the DNA that make it red, that make it fluoresce red. And in the normal cells, you basically do exactly the same thing, but you make it fluoresce green. And then you bind it to a slide, right? Now this slide is why they call it an array comparative genomic hybridization, because believe it or not, what is on this slide is the entire human genome. So the human genome has about 3 billion DNA bases in it, 3 billion. But you can spot DNA from you can break it up into chunks of about 100,000 base pairs. And each one of these little tiny dots on this slide have about 100,000 base pairs of DNA from the human genome. And what has happened is it's in sequence. So they put the entire genome in blocks of 100,000 base pairs onto a single slide. And then what they do is they put the red DNA from the possible melanoma and the green DNA from the normal DNA. And when you look at them together, you can see it looks red here, it looks green here, but what you can tell when you overlap them is you can tell whether it's more red, like this spot, or more green, like one of these spots, or more yellow, because when you add green and yellow light, it means they're about equal. right? So if you have a red spot, that means that you've got more of that kind of DNA in that bad melanoma, possible melanoma, in that part of the genome. In that 100,000 base pairs, you've got more of that particular gene in the melanoma, the possible melanoma, than you do in the normal. Whereas if it's green, then you've got more. That means you have less of the red. And if you have a yellow color, then it's about the right amount. That means they're about the same. So again, if you have red color, it's more copies in the melanoma. If you have a green color, it's fewer copies in the melanoma. And if it's yellow, it's about the same number in the normal and the melanoma. I shouldn't call it a possible melanoma, I should say. Now you can quantify this because all of this light can be measured. And so you can see that these are all the yellows right here along the line where they're about the same. And this is where there's too much. These would be red, and this is where there's too little. So it would be green. And here's the actual picture that the pathologist sees to make the call. So what you see here is this is a regular mole. It's called a melanocytic nevus. This is just a regular, normal appearing mole. And what you can see is that the normal control DNA was probably, what we have here is we have 2 copies of an X and no copies of a Y. So that means this came from a female patient, but all the rest of the genes in chromosome 1, 2, 3, all the way through all of the chromosomes, everything else was at about the right level with the normal control DNA, except for you had 2 copies of X and no copy of Y. Now, in a melanoma, what you see is there are several places where you have either 2 copies or 1 copy, and maybe 3 copies here of this one. And what that suggests, especially since this is in chromosome 6, remember that was an important one, and one that's really important is 9p, which is right here. It's very commonly lost in melanomas. And so you can start to see that indeed, this pattern is able to show you that this is more likely to be a melanoma than it is to be a nevus. So CGH identifies if a melanoma has too many or too few copies in any gene, not just the 4 that were in fish. Now you can look across the entire genome. It's objective because you can quantify it. It is informative, incredibly informative, but it still doesn't tell you what mutations are there or even always prove it's a melanoma, right? Because how many of these changes do you need before it's an actual melanoma? There's still an indeterminate category. So let's keep going. Let's go to the gene expression profiling, or GEP test, right? Gene expression, so to understand where it's coming from, let's just review a minute about where we've been with fish and with CGH. So fish was looking at the DNA in the nucleus, right? And CGH was looking at the DNA. But the gene expression profile test is actually looking at RNA. And remember that RNA is a copy of the DNA. It's not actually the place where the DNA was caused to be. It's further from the cause of the problem, but it's also closer to the function. So when you have more RNA made, you usually get more protein, and that causes the abnormal pattern of behavior of the cell through the protein. If you have more RNA, you get more protein. If you have less RNA, you get less protein. So measuring the RNA is a way to tell whether or not there's a normal or abnormal amount of these really important proteins there or not. And so what you do with the gene expression profile test is you take the biopsy of your mole, right? And then you take the tissue and you remove the RNA now, not the DNA, the RNA, and you measure how much of a panel of RNAs there are. So you can't test every RNA, and this gene expression profile test is a profile. It's not the whole entire genome. It's just selected RNA molecules that are known to be associated with melanoma. And so you measure how much of the RNA of a whole set of about 20 to 30 different genes that are known to be important. And you then compare the profile of the expression of those 20 to 30 genes. You compare it to moles and you compare it to melanomas, and then you determine a probability of malignancy based on whether it's more similar to the moles or more similar to melanomas. And so let's go through that in a little different way. So you take RNA, that the way that these were developed, that these assays were developed, is that RNA was extracted from many melanomas that were clear melanomas. And they took that RNA, and then what they did is they compared it to the RNA from many, many benign moles, right? And the thing that's interesting is they did not use the indeterminate. They wanted to make sure that they knew absolutely, was it melanoma or was it not melanoma? And then they said, okay, these are the genes that we think are going to be the best markers to determine if those levels change, that it's either a melanoma or it's not a melanoma. So now you take the possible melanoma that we're talking about, that we looked at from the beginning, and you say, where does the expression of those RNAs, those 20 to 30 RNAs, which pattern does it look most like? Does it look like a melanoma, or does it look like a benign mole? And that's where you get even more information about whether or not your questionable mole is really a melanoma or really a mole. So gene expression profiling identifies if an indeterminate mole has RNA levels in 20 to 30 genes that are similar to melanomas or more similar to moles. It's objective, informative, but it doesn't prove it's melanoma, and there is still an indeterminate category where it's not a pattern that looks classic for either melanoma or moles. So I want to make some final clarifications, and then we're going to sum up. So for molecular testing, I think one of the things that's important for RNA, there's a lot there's two types of RNA gene expression profile tests that are out there, that are on the market, that people may have heard about and may have had run on their suspicious lesion. There's a diagnostic marker, which is the one I was just talking about, but there's another kind of gene expression profile that's called a prognostic marker. So with diagnosis, remember what we've been talking about is it a mole or is it a melanoma? Prognosis assumes that you've already put it into a category of melanoma. You don't use a prognostic gene expression profile test on a potential mole, because it's not gonna work. It's only telling you whether the melanoma, once you know it's a melanoma, whether the melanoma is likely to spread and kill or not. So those are two different things, right? So in the diagnosis, there's two different kinds of JEP tests out there now. There's one that uses a tissue block that cuts out part of the tissue itself. And there's another one that is testing cells that are pulled off of the mole by using tape stripping. But both of them are diagnostic. They're trying to determine whether it's a mole or whether it's a melanoma. One more set of clarifications, I think, just to sum up. Let's go back. We have FISH and CGH that show how many copies of the DNA are in the cells. Too many copies or too few copies is both abnormal, but it doesn't determine if the copies are doing anything bad for the cell. It's not associated with the function of the protein, and it's not 100%. Gene expression profiling shows that there's an abnormal amount of RNA from particular genes that are really important in causing melanoma. So it's getting closer, it's closer to the function, but it still doesn't explain why. And so there's overlap between acceptable levels. Every melanoma, again, unique, and there's still an indeterminate zone. It's just a smaller indeterminate zone. So you might say, well, what have we accomplished with all of this science, all of this technology? What in the heck? We still have an indeterminate zone. But I want to point out to you what's happened, because in ancient history with respect to melanoma, all you had was a great exam. And the great exam kept you really cannot tell the probably benign from the probably malignant by an exam. We've got dermoscopy now. We've got confocal microscopy that's narrowing that indeterminate zone even further. We've got biopsies with special stains now that are helping us to narrow the indeterminate zone. We've got FISH and CGH with DNA, and we've got gene expression profiling with RNA, and all of these are basically whittling down that indeterminate zone so that we were becoming more and more confident that we've accurately decided whether or not this was a melanoma or not. This, I just want to point out, I'm going to end where I sort of started, is that this is basically the macroscopic naked eye. This is the microscopic cellular level, and this is the molecular level. And it takes all of them when you run into a problem with indetermination to figure it out. And I'll end, this is my last slide, I promise. Together, we can eliminate melanoma as a cause of death. So join us. We hope you'll join us. These are my acknowledgments for all of the amazing people that have helped us. And I thank you very much for your attention. And I hope you have some really good questions. I'm excited to answer them.